You know that slimy sort of film you sometimes find, like in standing water?
Hmm.
Believe it or not, that's actually a type of biofilm. And get this, according to the NIH, biofilm is implicated in something like 80% of all infections.
That's a staggering number. It really is.
Okay, so let's unpack this because today we're diving deep into the fascinating and really crucial world of wound bioburden and infection.
Yeah, and what's really interesting is how our understanding has shifted. We used to think about wound infections mainly in terms of individual sort of free-floating bacteria.
Right, just loose germs.
Exactly. But now we realize these complex communities, these biofilms, they play a huge, often dominant role.
Precisely. And to help us navigate this microscopic landscape, we're drawing on insights from the chapter Wound Bioburden and Infection. It's by Nancy Stotts and Gregory Schultz from Wound Care Essentials, Practice
A
Principles.
really comprehensive resource.
Definitely. Think of this as like your shortcut to understanding a really vital part of how wounds actually work or sometimes why they don't work.
And our mission today is really to go beyond that simple idea of just germs in a wound. We want to explore the different stages microbes go through in a wound.
All right.
Understand why it's often so tricky to tell if a chronic wound is, truly infected.
Yeah, that seems key.
And look at the latest strategies for dealing with these complex situations. We'll be using this chapter as our main guide.
Great. So let's start with something pretty fundamental. This hidden world of microbes that actually live with us all the time. It turns out we're in this constant sort of balance, right?
That's absolutely right. Our skin, for instance, it's not sterile at all. It has its own complex ecosystem, the skin microbiome.
Like a little rainforest on your arm.
Kind of. And it's different everywhere. The microbes on your hand aren't the same as on your foot, and they vary from person to person, too.
And our skin isn't just sitting there, is it? It fights back.
It does. It has chemical defenses, oils, sweat that help keep unwanted microbes out.
Mhm.
But when a wound happens, it's basically an open door. Microorganisms can get in from all over our own skin, maybe the GI tract, the environment around us.
So this leads to the idea of bioburden. What is that exactly?
Bioburden is simply the number of living microbes on a surface. That could be wound tissue or even, say, a medical device.
Okay.
And for a long time, the focus was mainly on what we call planktonic organisms.
Planktonic, like in the ocean.
Sort of the same idea, yeah.
Yeah.
They're individual bacterial cells just floating around freely.
And that's what antibiotics were designed for, right? These free floaters.
Primarily, yes.
Yeah.
Antibiotics target specific functions in these individual cells to kill them or stop them growing. But here's where the game really changed.
Yeah.
Our understanding took a huge leap when we recognized the critical role of biofilm.
Okay. The biofilm again. So what happens? How does it form?
Right. It starts when these planktonic, free-floating organisms land on the wound surface. They start to settle,
Okay.
attach themselves. And then something really remarkable happens. They actually change their genetic expression. They switch to what's called a biofilm genotype.
They change their genes.
Wow,
Oh.
they do, to adapt to living in a community. And then they start producing this slimy substance called EPS, extracellular polymeric substance.
EPS. Okay. What's that doing? Is it like glue?
It's more like a scaffold or maybe like mortar holding bricks together. It's this hydrated matrix, mostly made of complex sugars, polysaccharides, but also proteins, fats, even DNA.
DNA in the slime.
Yep, extracellular DNA. And this EPS is absolutely vital for the biofilm because it acts like a protective shield
Ah, protection. From what?
from, well, pretty much everything trying to harm them. Antimicrobial drugs, disinfectants, and even the body's own immune cells.
So it's like they build a fortress around themselves.
Exactly like a fortress.
And
And the organisms inside, they're not just hiding passively, they actually communicate.
communicate. How?
They use chemical signals. It's a process called quorum sensing. It allows them to coordinate their behavior.
Like sending little chemical messages.
Precisely. They work together, they change their metabolism, often slowing it right down. And this is really crucial. They become much, much more tolerant to antibiotics.
Okay, now the chapter makes a really important point there, the difference between tolerance and resistance. Can you break that down?
Absolutely. It's a key distinction. Resistance is a permanent genetic change in the bacteria. A resistant bacterium is resistant whether it's floating free or in a biofilm. It's hardwired.
Okay.
Tolerance, on the other hand, is temporary. It's the ability to survive antibiotics only when the bacteria are living inside that protective biofilm structure.
Ah, so if you broke up the biofilm and they floated free again?
They'd likely become susceptible to the antibiotic again, assuming they aren't also genetically resistant, of course.
So the biofilm environment itself is creating this tolerance. How?
Several ways. The dense EPS matrix can physically block antibiotic molecules from getting deep inside to reach all the bacteria.
Like a barrier.
Exactly. Plus, many bacteria within a mature biofilm significantly slow down their metabolism. They become less active. And since many antibiotics work by targeting active processes like cell wall building or protein synthesis,
If the bacteria aren't doing much, the antibiotic doesn't have much to target.
you got it. It's like trying to disable a machine that's already switched off. It's much harder.
That makes a lot of sense. And are these biofilms usually just one type of bacteria hanging out?
Usually not, actually.
What?
More often than not, they're polymicrobial.
Meaning multiple types.
Yeah, different species, sometimes even fungi, all living together in this coordinated community. They even create these tiny fluid-filled channels inside the biofilm structure.
Like little plumbing.
Sort of.
Yeah.
It acts like a mini circulatory system,
Sure.
letting nutrients and oxygen in and carrying waste products out.
Wow. And you mentioned the slower metabolism. Does that have other effects? Like they can go dormant?
Yes, exactly. This low metabolic state lets them conserve energy and basically just wait, like hibernating. Then, if conditions become more favorable, they can reactivate and start multiplying again.
And that dormancy makes them even harder to kill with antibiotics, I guess.
Much harder. Because again, the antibiotics often need the bacteria to be actively growing or dividing.
So it really sounds like biofilm is a massive roadblock to wound healing.
It absolutely is. By shielding the microbes from the immune system and constantly triggering low-level chronic inflammation, biofilms can essentially lock a wound in a non-healing state.
And it doesn't stop there, right? They can spread.
That's right. Biofilms can periodically shed individual planktonic bacteria or even small clumps of biofilm back into the wound environment.
Which can then drift off and start new biofilms somewhere else.
Exactly, perpetuating the cycle. And even bits of the EPS matrix itself can cause problems. That extracellular bacterial DNA, the ebDNA we mentioned, it's inherently inflammatory. It can directly activate the host's immune system, just adding fuel to the fire.
This paints such a complex picture. It's not just germs, it's like a whole city with defenses and communication. Okay, the chapter also talks about something called the wound bioburden cycle. What's that describing?
It describes the sort of predictable progression, the stages of how microorganisms interact with a wound. It can lead either to the body successfully clearing them out or eventually to infection.
And what determines which way it goes?
It's a combination of things. A person's own immune system and overall health are huge factors. Then there's the local environment in the wound itself. Is it moist, warm? What's the pH? Is there enough oxygen? And finally, of course, the amount and the aggressiveness, the virulence of the microbes present.
Makes sense. And there are 4 main phases in this cycle. Let's walk through those.
Okay. First is contamination. This happens right after injury. Microbes, bacteria, fungi, maybe viruses just land in the wound from various sources.
So just their presence.
Exactly. They're present, but usually free-floating, planktonic. There are no real signs or symptoms of trouble yet, and this is often the body's best chance to clear them out with its natural defenses.
Okay, contamination. What's next if they aren't cleared out?
Then we move to phase 2,
Sure.
colonization. Here, the microorganisms start to stick to the wound surface. At first, it's kind of a loose attachment, reversible. They can still be washed off fairly easily.
But then they dig in.
Right, they become more firmly attached. They start using the wound bed for food, and they multiply. Early biofilm structures might start to form, but crucially, still usually no visible signs or symptoms of infection.
Okay, so they're setting up shop, but quietly. What's phase 3?
Phase 3 is critical colonization biofilm. This is where things really start to change. The biofilm genotype, that genetic shift, is established. The EPS layer gets thicker. And now we start seeing problems.
Like what?
Localized signs. Delayed healing becomes noticeable. There might be a low-level local infection brewing. The chapter really stresses that this phase reflects the impact of that developing biofilm.
So the key insight here is that delayed healing itself could be a sign of this critical colonization stage, a sign of biofilm building up.
That's a huge takeaway, yes. Delayed healing isn't just stuck. It could mean significant bioburden, likely in a biofilm.
Okay. And the final phase?
Phase 4 is outright infection. Now, the organisms aren't just sitting on the surface, they're actually invading the living tissue around the wound. This triggers the classic systemic signs of inflammation we usually think of as infection redness, heat, swelling, pain, maybe increase pus or exudate.
And this is when antibiotics might work, but mainly on the active bugs.
Yes, primarily against the metabolically active ones. The ones hiding deep in the biofilm or the dormant ones might still evade the treatment.
In the chapter mentioned, there's sort of an alternative, maybe more modern way to think about this cycle, too.
Yes, reflecting our better understanding of biofilm. Some researchers propose a slightly different sequence. Contamination, then attachment, then proliferation, multiplying, then reaching a critical concentration of those quorum-sensing signal molecules.
The chemical message.
Right. Followed by the adaptation of the biofilm phenotype, that genetic switch, and finally the establishment of a mature, often polymicrobial, biofilm. It's a more nuanced view, highlighting the biofilm's development.
It really shows how dynamic things are in a wound. Now, you mentioned the body's defenses earlier. How does our immune system normally handle these microbes? And crucially, when does it get overwhelmed?
Well, when you get injured, the first thing that happens is inflammation. It's a vital response.
Mm-hmm.
Immune cells, primarily neutrophils and monocytes, flood the area.
The first responders.
Exactly. Neutrophils arrive almost instantly, engulfing bacteria. Then monocytes arrive and turn into macrophages, which are like the cleanup crew, but also fight microbes. This whole process of engulfing and destroying microbes, phagocytosis, needs oxygen.
Ah, so good blood flow and oxygen are critical for our defenses to work properly.
Absolutely critical. Along with the cells, the body releases inflammatory signals, reactive oxygen species, which are like chemical weapons against microbes, enzymes called proteases, and antimicrobial peptides, all designed to kill the invaders.
And in a simple acute wound, this all calms down fairly quickly.
Usually, yes. In an acute wound that's healing normally, this intense inflammation resolves within about 5 days. The neutrophils die off, macrophages clear up the mess, and healing progresses. Infection is prevented.
But chronic wounds are different. The inflammation just keeps going.
Precisely. That's a hallmark of chronic wounds' persistent, non-resolving inflammation. The immune cells keep coming, keep releasing those damaging substances like reactive oxygen and proteases. And sometimes the bacteria themselves contribute, like Pseudomonas producing rhamnolipids that help them avoid being eaten by immune cells.
And those proteases become a problem because they don't just attack microbes, they start breaking down the good stuff, the extracellular matrix that's needed for rebuilding tissue.
Exactly. It creates this destructive cycle. The chronic inflammation and the proteases damage the host tissue and stop healing, while the biofilm protects the microbes, allowing them to persist and potentially cause a full-blown infection.
And that explains the huge difference in infection rates we see.
It's a major factor. Less than 2% infection rate in many acute surgical wounds, compared to estimates as high as 53% in chronic wounds. It really highlights the challenge.
A massive difference. Okay, so given all this complexity, how do we actually define when a wound has crossed the line from, say, critical colonization into being truly infected?
Well, the standard definition of wound infection involves microorganisms invading and multiplying within living wound tissue. And this invasion has to cause some kind of harm or tissue injury.
So not just sitting on the surface, but getting into the viable tissue.
Exactly. Another trigger for defining infection is simply finding beta-hemolytic streptococcus, because it's known to be particularly aggressive.
Yeah.
So the key parts of the definition are viable tissue, organism invasion, and multiplication, the host defenses being overwhelmed, and usually visible signs and symptoms.
What about pus? Doesn't pus mean infection?
Not necessarily on its own. Pus is mostly dead white blood cells and debris from the fight. While it's often present in infection, you can have pus without a true invasive infection. And importantly, infection can't happen in dead tissue, like thick slough or necrotic eschar. But
that dead tissue is like a 5-star hotel for microbes.
absolutely, it provides food and shelter,
Yeah.
helping them multiply and potentially invade the healthy tissue nearby. So removing it is crucial. The key thing for you to remember is that visible signs like pus don't automatically equal infection, especially in chronic wounds.
So this traditional definition, it sounds like it was mostly developed with acute wounds and those free-floating bacteria in mind.
Largely, yes. Now, with our understanding of biofilm being involved in most infections, particularly chronic ones, there's a real question. Should that critical colonization biofilm phase, the one causing delayed healing, be considered and treated as a form of infection?
That's a good question.
The hope is that as we get better tools, maybe bedside tests, to directly measure bioburden and biofilm, we'll get a clearer picture and target treatment better.
But for now, diagnosis still leans heavily on those signs and symptoms, right? Which, as we said, can be tricky.
Very tricky, especially distinguishing infection from the normal inflammation you see in the first few days after any injury.
So how do clinicians manage?
Well, for acute surgical wounds, there are established criteria. The CDC's surgical site infection, SSI criteria. They're quite specific. But chronic wounds are a different beast.
Because they often have that background inflammation already. And biofilm.
Exactly. The signs of a new infection can be much more subtle or masked by the existing state. And there's no single universally agreed upon set of criteria for chronic wound infection.
Which leads to inconsistency.
It can, yes. Inconsistent diagnosis, maybe inconsistent or delayed treatment,
Right.
and we still lack routine clinical tools to directly assess how much biofilm is contributing.
So if the classic signs redness, heat, swelling, pain aren't always reliable in chronic wounds, what should clinicians be looking out for? Are there more subtle clues?
There are. Researchers like Cutting and Harding identified some additional signs often seen in chronic granulating wounds. These include things like the wound suddenly stops healing or gets worse.
Okay, stalled healing.
Right. Changes in the color of the granulation tissue, maybe it looks dusky or unhealthy. Tissue that bleeds very easily, we call that friable.
Friable granulation tissue.
Unexpected or increasing pain is a big one. Finding new pockets or tunnels under the wound surface. Abnormal smell. An actual breakdown or deterioration of the wound.
And their research found that even experienced clinicians didn't always agree when assessing these signs.
That's right, highlighting the need for better education and consistent use of these specific chronic wound criteria. Then, a really important study by Gardner and colleagues dug deeper.
What did they find?
They found that these chronic wound-specific signs, particularly increasing pain, friable granulation tissue, foul odor, and wound breakdown, were actually better predictors of infection in chronic wounds than the classic signs.
Better than redness and heat.
In their study, yes. And increasing pain and wound breakdown were especially strong indicators. If they were present, infection was very likely.
That's really practical information. And then we have those acronyms, NERDs and STONIES, that try to capture these signs.
Yes, developed by Sibold and colleagues. NERDS helps identify suspected critical colonization, or biofilm, it stands for. Non-healing, exudate increased, red and friable tissue, debris, slow, smell bad.
So if you see several of those, think biofilm.
The idea is if 3 or more are present, it's highly suggestive. Then STONIES points towards a deeper, more serious infection. That's size increasing, temperature increased, local heat, osseous probe to bone suggesting osteomyelitis, new areas of breakdown, erythema, redness, swelling, spreading, exudate, pus-like, smell bad.
Again, 3 or more suggests a deeper problem.
Correct. Studies trying to validate these have shown some correlation with swab culture results, but there are limitations. They're surface assessments, they don't measure deep tissue burden directly, and they don't specifically confirm biofilm.
Right. Still useful tools, but not definitive.
Exactly. Other research, like expert consensus panels, has reinforced many of these signs, things like cellulitis spreading, bad odor, pain, delayed healing, wound getting worse, increased exudate.
And a review by Reddy's group looked at how well these signs stack up against the gold standard tissue biopsy.
Yes, and they found that, based on the available evidence, increasing pain seemed to be the most helpful clinical sign suggesting infection, but its absence didn't reliably rule infection out.
So pain is a clue, but lack of pain isn't proof it's not infected.
Right, and they confirmed that the classic signs often aren't very helpful in chronic wounds. A major issue they highlighted is the lack of high-quality studies directly comparing signs and symptoms with biopsy results. Plus, many signs are subjective.
Like smell or how friable the tissue looks.
Exactly. How reliably do different people judge those? Often we don't know. Reddy's team felt Gardner's research provided the strongest evidence currently available. It's also crucial to remember, biofilm itself isn't usually a visible sign. We don't have a routine test for it, and it can look just like SLA or normal exudate.
But an international group of biofilm experts did identify some indirect clues, right? Things that make you suspect biofilm.
Yes, they agreed strongly on a few things. One big clue is treatment failure. If a wound doesn't improve, even when you use antibiotics or antiseptics chosen based on a culture.
That points to biofilm.
Very strongly, yes. Other potential clues include recurring flare-ups of inflammation, consistently high amounts of exudate, and maybe seeing a gelatinous, slimy layer on the wound that comes back quickly after you wipe it away.
Interesting. What about delayed healing?
They had weaker agreement on whether delayed healing alone is a specific sign of biofilm. The main takeaway was, you often infer biofilm is present because your standard treatments aren't working, rather than seeing something specific like SLA or exudate.
Okay, so clinical signs can be really ambiguous. What about diagnostic tests? How can we get a better look inside the wound's microbial world?
Well, the long-standing gold standard for diagnosing wound infection is a tissue biopsy for quantitative culture.
Taking a small piece of tissue.
Exactly. Historically, finding more than 100,000, that's 10 to the fifth, power colony-forming units, CFU, of bacteria per gram of tissue, or any amount of beta-hemolytic streptococcus, was considered indicative of infection.
And that number, 105, came from studies looking at things like skin graft success.
Yes, studies correlated bacterial levels above that threshold with things like graft failure or delayed wound closure in acute wounds. However,
There's always a however with biofilm, isn't there?
there usually is. We now recognize that standard cultures, even from biopsies, primarily grow the planktonic bacteria. They likely underestimate the total number and diversity of organisms, especially those tucked away in biofilms.
And biofilm isn't spread evenly, so one biopsy spot might miss it.
Precisely. A biopsy from one area might not represent the whole wound. And again, no routine clinical test specifically identifies biofilm bacteria within that tissue sample. So even the gold standard has limitations in the biofilm era.
So biopsy is our best bet, but it's not perfect. What are the other options for culturing?
Besides biopsy, the main methods are aspiration, using a needle to draw fluid and swab cultures.
Oh, okay.
Each has pros and cons regarding invasiveness, accuracy, and the type of information you get. And regardless of the method, getting good clinical information on the lab slip and getting the sample transported quickly is vital.
And the lab can process these in different ways, like just seeing what's there or counting them.
Right. Qualitative just tells you what species are present. Quantitative gives you an actual count per gram or milliliter. Semi-quantitative is common. They dilute the sample and plate it to estimate the amount, often reported as 1 plus to 4 plus 12. A 4 plus result is often used as a cutoff, suggesting infection.
And they can test which antibiotics work.
Yes, antibiotic sensitivity testing is usually done. And you need to specifically ask for anaerobic or fungal cultures if you suspect those, as they require different lab techniques.
Okay, let's quickly compare these. Biopsy. Gold standard, but invasive, painful, needs expertise, might miss patchy biofilm. What about aspiration?
Aspiration has less research behind it. Some data suggests it might underestimate infection compared to biopsy, and the bugs you find in the fluid might not be the same ones deep in the tissue. So it's not generally recommended for routine culturing, but can be useful for sampling an abscess, like a collection of pus.
Makes sense. Which leaves swabs. Probably the most common method.
Definitely the most common, because they're least invasive. There's been a lot of study on swabs. Early work by Levine described a specific technique that seemed to correlate better with biopsy results.
The Levine technique, how does that work?
You firmly press and rotate a swab over a 1 centimeter squared area of clean wound bed for about 5 seconds, aiming to collect fluid from within the tissue, not just surface gunk.
As opposed to just wiping the surface, like with the Z-stroke method.
Exactly. The Z-stroke just zigzags across the surface. Generally, studies suggest the Levine technique gives more reliable results that better reflect the tissue bioburden.
So what are the pros and cons of swabs overall?
Pro, easy, quick, minimal training needed. Con, mainly samples, surface organisms, might miss deeper or anaerobic bacteria. Risk of getting a poor sample. It's usually recommended to cleanse the wound first to remove contaminants.
So clean the wound, then swab using the Levine technique.
That seems to be the best practice based on current evidence. Moistening of the swab might help too. But a big caveat is that many older studies comparing swabs to biopsies didn't specify how the swab was taken, which makes interpreting them difficult.
But Gardner's group specifically compared techniques.
Yes, they found Levine technique swabs were more accurate and agreed better with tissue cultures than just swabbing exudate or using the Z-stroke. They even found certain bacterial count thresholds on Levine swabs that predicted higher tissue counts. A systematic review also favored Levine over Z-stroke for initial monitoring.
But biopsy is still better for tricky cases.
Generally considered more reliable, yes, especially if you suspect antibiotic resistance so the wound isn't responding.
And what about more recent comparisons?
A study by Hall, Boom, and colleagues directly compared Levine swabs to punch biopsies in chronic wounds. They found surprisingly similar results in many cases, especially if they ignored common skin contaminants.
So maybe swabs aren't as bad as we thought, if done correctly.
Their conclusion was that starting with the less burdensome Levine swab makes sense and reserving biopsy for wounds that don't respond to treatment based on the swab results. But they still stressed, neither technique truly measures the total bioburden, especially biofilm.
Okay, so evidence-based practice points towards using the Levain technique for swabs. But even then, surface cultures are maybe best for initial monitoring, while biopsy gives the most accurate picture, despite its own limitations. Are there newer techniques on the horizon that might help specifically with biofilm?
There's a lot of research happening. People are using advanced microscopy, sophisticated culture methods, and molecular techniques like PCR or sequencing looking for bacterial DNA like 16S RNA.
To get a more detailed map of the microbes.
Exactly. These can identify more organisms, sometimes faster, and even give clues about the biofilm structure. But most are still expensive and mainly used in research labs. And a key limitation of many molecular tests is they detect DNA. They can tell you a pathogen is present, but not necessarily if it's actively causing problems or if it's in planktonic versus biofilm form.
So useful for identification, but not the whole picture. Any other specific things being tested?
Yeah, the chapter mentions blotting, pressing a special membrane onto the wound, then staining it. Early work suggests this might predict SLU development, possibly linked to biofilm. Also, point-of-care protease detectors.
Measuring those damaging enzymes directly in the wound fluid.
Right. They're in development, approved in some places, but not the U.S. yet. That could give real-time information about wound inflammation and bacterial activity.
It really sounds like better biofilm diagnostics are urgently needed. We lack a simple bedside test right now.
That's a major gap, yes. Improving diagnostics is a huge focus of research.
Okay, let's shift gears then. If diagnosis is tricky, how do we approach treatment? Especially for these complex wounds with high bioburden or biofilm.
Well, the foundation is good wound bed preparation. That time paradigm is key. Tissue management, infection inflammation control, moisture balance, and edge advancement for epithelialization.
So prepping the wound bed properly.
Yes, but it's holistic too. Treat the whole patient, optimize their health, oxygenation, blood flow, nutrition. Address the underlying cause of the wound. Recognize that chronic wounds are different. They have defective tissue matrix, prolonged inflammation, often high bioburden. The goal is to shift the chronic wound environment back towards an acute healing state.
And you mentioned a specific model for tackling biofilm, the step-down, step-up approach.
Right. This is a comprehensive strategy proposed for chronic wounds, wounds healing by secondary intention, and infected wounds. The core idea is early and aggressive intervention.
Aggressive how?
It involves several things bundled together right from the start. Thorough debridement is number one. Then potentially using empiric antibiotics, both systemic and topical antiseptics, while waiting for diagnostics. Managing the patient's overall health. Trying to identify the organisms early, ideally including biofilm bacteria if possible.
And then adjusting based on response.
Exactly. Constant reassessment. Are the antiseptics or antibiotics working? Do we need to change them based on culture or molecular results? Are biofilm disruption techniques needed? If the wound stalls or worsens, you step up therapy, maybe adding adjunct treatments. If it improves, you can eventually step down to standard care.
So it's proactive and adaptable.
Precisely. The aim is to hit it hard and early, ideally before planktonic bacteria can establish a mature, hard-to-treat biofilm. How it's implemented depends on resources, of course, especially access to things like molecular diagnostics.
And there's a rough timeline suggested.
Yeah, the model suggests in the first few days, 1 to 4, you combine therapies, debridement, empiric antimicrobials, biofilm disruption, managing host factors, maybe getting samples for DNA analysis. Days 5-7, you start personalizing based on wound status and initial diagnostics, maybe switching to targeted topicals, continuing host factor management. Then weeks 1 to 4 involve ongoing reassessment and adjustment.
Debridement keeps coming up as step 1. Why is it so critical when dealing with bioburden and biofilm?
Because it removes the stuff microbes love. Devitalized tissue is food and shelter. And crucially, debridement aims to physically remove existing biofilm and the biofilm genotype organisms. The big challenge. Biofilm is often invisible, and even sharp surgical debridement probably doesn't get all of it.
But it disrupts it, at least.
Yes. And sharp debridement is vital for getting into tunnels and undermined areas where biofilm can hide. Plus, after a good debridement, you create this window, maybe 24 hours, where topical agents can work better on any remaining planktonic bugs before they reattach.
Because biofilm can reform quickly, right?
It can start reforming within 72 hours. That's why regular debridement, at least weekly sharp debridement, is often recommended as a minimum for chronic wounds with suspected biofilm.
And sometimes this needs to be done surgically.
Yes, surgical debridement in an operating room allows for better pain control, managing bleeding, and potentially removing more tissue more thoroughly. But the key is having trained personnel who know how and when to debride sharply. Referral to a wound center might be needed.
Are there other debridement methods useful against biofilm?
Well, besides sharp surgical, there's hydrosurgery, ultrasound, coblation. Evidence is still emerging for some of these, though ultrasound might reduce exudate and improve healing times in some studies. Monofilament pads offer a mechanical way to remove SLU. Enzymatic debridement is slower, less traumatic, but its value against biofilm isn't really established.
What about maggots, biodebridement?
Maggots, sterile larvae, definitely help debride. They also seem to have some anti-inflammatory effects. Studies show they can debride faster than some other methods initially, but effects on overall healing time and pain are variable. Some reviews suggest they might improve healing outcomes. Often they're used in combination with other methods.
So sharp debridement seems central, maybe combined with other techniques.
That's often the approach, yes.
Huh.
Thorough removal of non-viable tissue and disruption of biofilm is paramount.
Okay, after debridement, what about cleansing and irrigation? How do they fit in?
They're important adjuncts, used after debridement and with dressing changes. The goal is to wash away loose planktonic bacteria before they attach, remove debris, contaminants, inflammatory exudate. Basically make the wound less hospitable.
Does it matter what you cleanse with? Saline, tap water, special cleansers.
That's debated. Some big reviews found no major difference in infection or healing between normal saline and clean tap water, or even tap water, versus no cleansing in some cases. But safe tap water is a key assumption there.
So saline is generally safe and simple.
Yes, and easily made at home if needed. Commercial cleansers with surfactants might help lift debris better in very exudative wounds. But you have to balance cleaning power with potential toxicity to healing cells. Skin cleansers are generally too harsh for open wounds.
So cleansing is recommended, but the best agent isn't clear. What about irrigation pressure?
Irrigation uses fluid force. The optimal pressure is thought to be 515 psi. Lower might not be effective. Much higher could potentially damage tissue or spread bacteria in chronic wounds.
How do you get that pressure?
A simple way is a 35 milliliter syringe with a 19-gauge needle or angiocath that gets you around 8 psi. Pulsed lavage systems deliver pressure pulses and might be more effective in some situations, but evidence is still developing. Whirlpool is another option for cleansing large areas, but avoid direct jet impact.
So targeted irrigation at the right pressure, especially for exudative wounds, seems helpful. Let's move to topical agents. You mentioned they're back in favor.
Yes, their role is seen as essential now, especially for critical colonization, biofilm, and infection, given the challenges of chronic wounds, biofilm, and antibiotic resistance.
No, they were.
They're applied directly to the wound liquids, gels, and dressings. They aim to kill remaining planktonic organisms after debridement, prevent reattachment, and suppress biofilm reformation.
Should they be chosen based on cultures?
Ideally, yes. Empiric use might be necessary initially, but the step-down, step-up model suggests using diagnostics like DNA analysis, if available, to guide the choice of topical agent based on identifying the specific organisms and their sensitivities. Then you adjust as needed. The challenge is access to and interpretation of those advanced tests.
And the big issues are still effectiveness against biofilm versus planktonic bugs and toxicity to healing cells.
Exactly. The ideal antiseptic is a magic bullet, kills microbes, including biofilm. Low toxicity to us doesn't stop healing. But many agents affect both microbes and our own cells.
The chapter lists quite a few iodine, silver, PHMB, honey. Can you give us the quick version on those?
Sure. Iodine. Formulations like povidone iodine, PVPI, or cadexmer iodine are used. PVPI disrupts bacterial functions, effective against MRSA, can inhibit some biofilms in vitro. Cadexmer iodine releases iodine slowly, absorbs exudate, seems less toxic to healing cells, and has some clinical evidence supporting its use in venous ulcers.
Okay. Silver.
Silver ions disrupt bacterial proteins. Comes in creams, dressings. Silver sulfidazine cream showed some benefit in reducing bacteria in leg ulcers in some studies, but other reviews found no clear healing advantage for venous ulcers compared to non-silver dressings. Lab studies on biofilm effectiveness are mixed compared to iodine.
What about PHMB?
Polyhexamethylene biguanide. Broad spectrum, seems to have low resistance potential and good safety profile regarding healing. Some clinical trials show PHMB dressings reduce bioburden, pain, and wound size in chronic wounds.
And honey.
Honey has natural antimicrobial properties. Reviews suggest it might help heal infected surgical wounds faster than some standard treatments, but the overall evidence quality is often rated as low or inconsistent.
So topical antimicrobials are a key second step after debridement, but choosing the right one isn't always straightforward.
Not always. And with silver, for example, the amount and release rate from dressings varies hugely. Too much might actually slow healing by affecting our cells. Selection should be based on the whole clinical picture, not just the silver label. Good news is, significant resistance to these major antiseptics hasn't really emerged.
What about systemic antibiotics, the ones you take orally or intravenously? When do they come in?
They're crucial for clear, spreading infections, or if the patient shows systemic signs of illness. But antibiotic resistance is a massive global threat, so we need to be judicious.
Use them wisely.
Exactly. The step-down, step-up model advocates for identifying the organisms and tailoring therapy if possible. Systemic antibiotics might be started empirically for suspected infection, but getting specific sensitivity data early helps optimize treatment and limit resistance.
Does the type of chronic wound matter for choosing antibiotics? Like, do diabetic foot ulcers need different antibiotics than pressure injuries?
Interestingly, studies analyzing the microbiomes of different chronic wound types often find similar key players. Lots of Staphylococcus, Pseudomonas, often anaerobes. This suggests that the initial empiric antibiotic choice might not need to be drastically different based solely on the wound type.
So focus more on the severity and individual patient factors.
And combining anti-biofilm strategies, like debridement and topicals, with appropriate systemic antimicrobials seems key for chronic wound infections. Avoid routine or prolonged use. Forward therapy is usually reserved for deeper infections with systemic signs, and it only works if there's good blood flow to deliver the drug.
Right, delivery is key. This brings us back to host factors and wound-specific treatments. You stressed this earlier. Why is optimizing the patient so important?
Because the body needs the right conditions to heal and fight infection. You need a thorough assessment. Is blood supply adequate? Infection thrives in ischemic tissue. Is there swelling that needs managing? Is oxygen getting to the tissues? Are blood sugars controlled? High glucose impairs white blood cells. Is nutrition adequate?
Got it. Fueling the fight.
Exactly. And managing any underlying diseases contributing to the wound or poor healing. Plus, specific wound care like reducing edema in venous ulcers or offloading pressure ulcers in diabetic foot ulcers. If you don't address these fundamental host and wound factors, your infection treatments might fail.
Setting the stage for success. Okay, lastly, the chapter mentions adjunctive therapies. What are those?
These are extra treatments considered as part of that early aggressive approach, or if standard care isn't working. Examples include negative pressure wound therapy, NPWT.
Vacuum therapy.
Yes. It helps draw fluid out, stimulate granulation tissue, particularly good results seen in diabetic foot ulcers. There's also NPWT with instillation, adding an irrigant, which showed some promise against biofilm bacteria in one study.
What else?
Collagen dressings can help manage destructive enzymes, protease in the wound. Research is exploring agents that specifically target the biofilm's EPS matrix, trying to break it down and expose the bacteria.
Like dissolving the fortress walls.
Kind of.
Yeah.
And quorum sensing inhibitors, drugs that block the bacteria's communication signals, are another research avenue, hoping to disrupt biofilm formation and maybe make antibiotics work better.
And targeted therapy based on molecular tests?
Right. As mentioned, some studies suggest choosing topical or systemic agents based on detailed molecular diagnostics leads to significantly better healing rates compared to standard empiric choices.
So lots of potential tools to add to the arsenal.
Definitely. The picture is complex, but our understanding and options are growing.
Okay, let's try to boil this down. What are the key takeaways for you, the listener? Well, first, it's crystal clear that biofilm isn't just some footnote. It's a major player in why many wounds get stuck or infected.
Absolutely. And this means we need to shift how we think about and treat wound infection, especially in chronic cases.
Second, figuring out if a chronic wound is truly infected is complex. The classic signs aren't always reliable, and diagnosis often requires looking at more subtle clues and considering the whole picture, including treatment response.
And third, treatment isn't just about antibiotics anymore. It's a multi-pronged attack. Aggressive debridement is crucial, followed by appropriate topical agents, managing the patient's overall health, addressing the wound-specific needs, and considering systemic antibiotics and adjunctive therapies when necessary.
So thinking about all this, the intricate nature of these biofilm communities, the constant evolution of diagnostics and treatments, here's a thought for you to ponder. How might personalized wound care change in the next, say, 10 years? How could we get even better at targeting these microbial fortresses to achieve faster, more reliable healing for everyone?
That's the exciting frontier. Will we have rapid bedside biofilm detectors, therapies that specifically dissolve the EPS or block quorum sensing, tailored antimicrobial combinations based on real-time molecular analysis, It's a space to watch.
Definitely. Look, this deep dive has really only scratched the surface of an incredibly complex topic. We absolutely encourage you to explore the research cited in the chapter and discuss these ideas with healthcare professionals.
Because understanding the science behind wound healing really empowers you, whether it's for your own health or for caring for others.